Bom dia, boa tarde, boa noite aos internautas que nos acompanham neste programa Nanotecnologia do Avesso, no seu número 423. Fazemos este programa dedicado ao público não especialista, porque é este público que paga as pesquisas sobre nanotecnologia realizadas no Brasil. Também fazemos esse programa porque é aqui que nós podemos discutir os impactos da nanotecnologia no meio ambiente, na sociedade, na economia, na ética, no direito, na saúde, coisas que não são discutidas aonde se faz nanotecnologia no Brasil. Por fim, fazemos esse programa porque acreditamos que ser livre é ser bem informado e a divulgação científica contribui e colabora para o aprofundamento da democracia e da cidadania no Brasil. Hoje, conforme tínhamos anunciado, teremos uma importante entrevista, colaboração do professor Chada Markin, diretamente da New Western University, em Boston, Estados Unidos, a quem nós estamos aguardando aparecer aqui no nosso Skype. A Elizabeth já esteve conosco, já testamos aqui o Skype e agora vamos aguardar a chegada do Dr. Marcos. Ok, Elizabeth?
Hi there! Pat should just be joining you any moment.
Okay. I don't have your image, huh? Only the photo of the professor.
Sure. Can I put that on as soon as Chad joins?
Yeah.
Great. My apologies for the delay.
Enquanto aguardamos o professor Marcos, vou avisar os nossos internautas que nosso programa, no dia 16, próxima segunda-feira, será com o nosso colega Adriano Premedida, que está organizando o nosso 15º Seminário Internacional Nanotecnologia, Sociedade e Meio Ambiente. Então, na próxima segunda-feira, das 11 às 12 horas, nós vamos discutir o programa do nosso próximo seminário. Será realizado na cidade de Porto Alegre, de 20 a 22 de novembro, lá na Universidade Federal do Rio Grande do Sul, no Instituto Latino-Americano de Estudos Avançados. Será a nossa casa lá para a realização do nosso 15º seminário. Muito bem. Estamos aguardando o professor Markin.
Oh, it is live?
The video.
Sure.
Hello?
Yes.
Great.
Wait. I don't have. Yes. Now I have an image of Professor Mark.
I can't see anything.
Yes. How are you, Professor?
I'm doing well. How are you doing?
More or less. I would like to tell you, please introduce yourself about your working with nanotechnology.
Okay, I'm Chada Markin, and I direct the International Institute for Nanotechnology at Northwestern. It's one of the largest institutes of its kind in the world. Focuses on the development of nanoscience and nanotechnology for impact in almost all fields. We have a strong focus in the development of new tools that can impact both biology and medicine, trying to develop ways of using nanomaterials to detect very low quantities of different types of disease markers so that you can study disease in unique ways, you can track disease, and ultimately treat disease in unique ways. And we've also developed a variety of new materials that can be referred to as nanomedicines that are beginning to be used to treat patients for many different types of diseases. One of key discoveries in the lab is the development of what we call spherical nucleic acids. These are structures made by taking spherical nanoparticles made of lots of different types of materials and synthesizing short strands of DNA and RNA and assembling them on the surface of these tiny little particles into spherical or globular forms. And those structures turn out to have really interesting properties. And one of the properties they exhibit is that they will naturally enter many different cell and tissue types. So this has led to the ability to create new types of materials that can be used for treating diseases like psoriasis, where you can create creams that can go into the skin and begin to use genetic material to reverse the effects of disease. So we actually target inflammatory markers or genes that cause inflammation associated with psoriasis to reverse those types of effects. Those drugs are in clinical trials in Europe now, actually in Germany, and the early signs are really promising. Another type of drug is one that is designed to be systemically administered, and that is used to treat diseases of the brain, in particular glioblastoma multiforme, a lethal form of brain cancer. That also is in human clinical trials. That trial is being done at Northwestern right now, and the drugs are being administered to patients. In animals, we've seen these types of drugs have the unique ability to cross what's called the blood-brain barrier, localize in the brain and in the tumor of brain cancer patients, and in that case, we can selectively tweak or turn down genes that are associated with that disease, and in this case, keep the cells from dying so that we can selectively cause brain cancer cells to die and reduce tumor burden or eliminate hopefully tumor burden. And so that's really quite exciting. And then the third area is we're beginning to use the same types of structures as new and potent forms of immunotherapeutics, where we can use them to tweak a patient's immune system to begin to work in combination with different types of therapeutics to create ways of effectively training one's immune system to eradicate different forms of cancer. And now we're looking at lots of different types of cancers, ranging from lymphoma to melanoma to breast cancer to lung cancer.
And in this institute, how many people, how many research work there?
At the Institute, we have about 400 faculty involved and over 600 graduate students and postdocs involved.
And the budget, how much is the budget for this institute?
So all in, we've been going now for about 16 years, and we've raised over $1 billion US dollars worth of research and educational infrastructure.
And is public money or private?
Combination. So we rely heavily on Northwestern's initial investment, but also we have government grants from many different types of agencies, including the NCI, the NIH, National Science Foundation, all branches of the military, the Department of Energy, and a variety of others. In addition, we're very fortunate to have a lot of philanthropic support as well. So there have been many people that understand the power of nanoscience and nanotechnology to impact many subfields within medicine, but also the environment, the development of new materials to impact energy research. And so people have different passions, and they tend to realize that this is one of the fastest ways to creating new materials that can change certain fields and lead to major technological outcomes. So we've been very fortunate to have tremendous philanthropic support.
And you think in percentual, is 50-50, public, private, or how is the proportion?
No, I would say it's more about 80-20.
80-20.
80 on the public side, 20 on the private philanthropic side.
And you think that this is one budget sufficient to do that we make the projections for the future, or we have more money to do?
Well, I mean, look, it's all a matter of throughput and speed, right? We are very well funded, but to move into new areas, it often requires new investment. There is a constant fundraising aspect of what we do that goes along with the science and technology development. The interesting thing is, though, that nanotechnology to me is really a great gift to scientists, engineers, and doctors who are interested in frontier research, because it puts a lot of chemistry, physics, biology, and engineering problems in a package that many people can understand. I think most people get very excited about the field of nanotechnology. They feel like they know something about it because it's been popularized to some extent through the media and through the movie industry. And so, to me, it's a great gift in the sense that it's a common denominator for scientists and non-scientists. People feel like they understand something about it and want to be a part of it. And they know that it has a chance to really change almost every field. And so I think that's one of the reasons that we've been very successful in terms of getting so much engagement from the philanthropic side of things. And then when you couple that to the impact it can have in medicine, the whole field of nanomedicine, I think, is going to completely transform the general field of medicine as we know it today. And you're going to begin to see these types of technologies in medicines seamlessly integrated with our practice of medicine all around the world. And I think it's going to dramatically change how we track and ultimately treat disease in almost any form.
Okay, here in Brazil, the government cut the budget for science and technology area, cut it 50%. So it is a big cut. And the United States, what is doing?
I couldn't understand the last part.
Yeah, in the United States, occur the similar thing, or the federal government is not cutting the budget for science and technology?
Well, there's always a talk. Every budget in the federal government is under scrutiny. And so there's always talk about cutting and expanding in different places. I think the biggest thing that's happened in the last few years has been kind of a reallocation, how we effectively deploy the dollars that we have. The U.S. has invested substantially in science, and will hopefully continue to invest in science and technology development. And I think it's paid off. I mean, if you look at the U.S. and certainly our universities, they're some of the best in the world. People from all over the world come to train there. So it's a sign that we're doing something wrong. The only reason that these exist is that because we invest in science and technology research. What we learn through those activities not only trains next level of people that are going to work in many of the U.S. high-tech companies, but it also lays the foundation for the technologies of tomorrow. And so the other thing that I think is really interesting in the U.S. that's a major benefit from the investment that we have at the federal level is this entrepreneurial culture where people take early discoveries from universities and create startup companies and get them funded by various means, oftentimes investment from venture capital or companies or individuals or a combination thereof, and then take that science or technology to a point where they can either produce products themselves and distribute them all over the world, or where they partner with larger companies. But those partnering activities would not happen unless there was this middle group that translated the technology from the university to the commercial setting.
Okay, I think you can start your presentation for today about the new spherical nucleic ácido.
Oh, I was just going to talk to you about it. I don't have slides.
No, there is no problem. You can make a general presentation and I will put some questions.
Okay, I mean, as I said, I kind of did that in the opening. The interesting thing, I think one of the greatest discoveries out of the Institute is this very simple concept that if you take ordinary DNA and RNA, short snippets of it, which we now can synthesize at large scale using modern-day gene machines, and you use chemistry and nanoscience to rearrange it on the surface of a particle in the formation of a spherical structure, you get a new form of the most important molecule ever discovered by scientists, and that's DNA. And these structures, on a sequence-for-sequence basis, behave completely differently than the natural forms, the known forms, the ones that we studied, the prototypical duplex DNA structure that we're all familiar with. That Watson
It's very, between commas, it's very simple, the manipulation of DNA.
It's incredibly simple. Of course, it took a long time to learn the importance of that architecture, but that's the beauty of it, that you can take this molecule that is the blueprint of life, and you can rearrange it on the nanoscale into this globular form, and now it interacts with living systems completely differently. And that is, to me, just an unbelievable discovery, and we spent a long time trying to figure out why it interacts differently, and then more importantly, to figure out what we can do with those differences to create new types of nanomedicines.
And the security
Is what?
is security this manipulation of the DNA.
I still couldn't get the first word, is it?
I think it's danger or not danger to manipulate this DNA.
I still can't understand the question.
We talk about the DNA manipulation, okay?
Right.
You answer me that is easy to do this.
For some reason, you're not coming across very clearly.
That is easy to do this.
Is it easy to do this?
Yes. Okay.
Yeah,
But
it's...
is danger to do this or not?
Oh, it's dangerous.
Yeah.
No. It's in fact the exact opposite.
Yeah.
So by doing this, remember what we're doing is we're not manipulating your DNA, we're not manipulating my DNA. What we're doing is we're making a new synthetic structure, and we can produce at scale. We can use chemistry to make DNA, and we can arrange DNA in the spherical form on the surface of a particle. We can make large quantities of it. And those structures can become the basis for either probes that allow you to track biological events, or the basis for new types of medicines that can be used to correct disease. And so one of the re-emerging tools in the pharmaceutical industry that are coming down the pike have to do with nucleic acid medicines. So let me kind of step back a second. If you think about pharmaceutical development over the last several decades, we've gone through three waves of development. The first wave was based upon small molecules, the idea that you can discover a molecule like aspirin that can alleviate pain, and of course has all sorts of other benefits as well.
Yeah.
The whole field, the whole pharmaceutical industry started because of those types of capabilities, and so chemists and medicinal scientists began to look for all sorts of different types of small molecules that could become the basis for drugs. And for several decades, that was how we developed drugs. Over the last couple of decades, we've learned that other types of structures can be very good as well, very useful as well. Structures based upon what we call biologics, peptides or proteins, antibodies. These are structures that are made naturally and isolated and then used as drugs. They account now for 7 of the top 10 pharmaceuticals sold today throughout the world.
Yeah.
Humira is an example of a biologic, right? A very, very potent potent, very powerful drug. It's the biggest seller for AbbVie Pharmaceuticals. So now what we have are two waves that are growing concurrently, one based on small molecules, the other based on biologics. It's not a matter of one being better than the other. They're just both feedstocks or platforms that can begin to generate all sorts of new drugs. Many people believe that the next wave of pharmaceuticals will be based upon short snippets of DNA and RNA, where you can begin to use basically sequences of DNA and RNA that are made by chemical means and then used as therapeutics to reverse the effects of disease at its genetic roots. So the idea is that you can use short strands of DNA if you can get them to enter into cells and enter into certain pathways associated with disease. You can begin to reverse the effects of a cell, for example, that's gone haywire, that's gone wrong.
And how nanotechnology is important in this process?
Okay, so let me explain that. Now, so why would DNA and RNA-based medicines be so powerful? Well, they're potentially very powerful because, in principle, you can treat and maybe cure disease as we know it. Because through science, through biology, through medicine, medicinal research, we have learned many of the pathways for disease progression. And we know the genetic roots of those pathways. So the idea is that you can create a short snippet of DNA or RNA to go and interfere with those pathways and stop the disease progression from occurring, and maybe even reverse it. And you can do that in a very selective way because DNA is made of a sequence, right? You've got the A's, the T's, the G's, and the C's, the chemical code that makes up DNA. Yeah. It's very unique. And so we can synthesize a very, not only potent medicine based upon nucleic acids, but a very selective medicine, one that only hits what it's supposed to hit in terms of target, and therefore you have very few side effects in principle. That's the good news. So for this to become a reality, I have to be able to make DNA and RNA in large quantities. Over the last 20 years, we've learned how to do that. Number two is I have to understand all the pathways associated with disease, because I have to know what pathways to enter or reverse to correct disease. Well, the good news is with something called the Human Genome Project, scientists have discovered many of these pathways. And we're going to pretty soon know everything, which is really exciting. But the third area that's really important, if you don't solve this problem, none of the rest matters. You have to be able to get these medicines where they need to go to treat disease. They have to go into the right types of cells. And if you can't do that, it doesn't matter what you do in the other two categories. So it's those three categories that make the difference. So where does nanotechnology come into play? Take spherical nucleic acid discovery. The idea that you can take DNA and RNA and rearrange it into a globular form on the surface of a nanoparticle, and now it will enter almost any cell type it encounters. That's really powerful. That means I can create, for example, creams that I apply to the skin, and they'll go into the keratinocytes, the skin cells that make up your skin. And I can begin to correct diseases associated with the skin. For example, psoriasis, atopic dermatitis, melanoma, a wide variety of diseases with a known genetic basis. There are over 200 diseases of the skin with a known genetic basis. If I take the brain cancer example, if I systemically inject spherical nucleic acids, they go everywhere. But they also do what most drugs won't do. They will actively cross the blood-brain barrier, and they can localize in the brain. And that might be a scary thing to you, but that's actually a really exciting thing. Because what that means is, for somebody, for example, who suffers from glioblastoma multiforme, lethal form of brain cancer, I can systemically inject these structures. They will go to the brain and they will localize a tumor. And if I encode them with genetic material that will reverse the effects of brain cancer or cause brain cancer cells to selectively die in the presence of healthy cells, I can begin to, in principle, treat that disease in a way not possible today. And so we're beginning to test that hypothesis. But if you think about this, you said, where does it come to play? This allows us to get DNA and RNA in places that ordinarily won't go. We can put them in eye drops, and they'll go into your eye. And you can begin to reverse the effects, for example, of things like glaucoma, macular degeneration, I can use them in inhaled form and take them into the lung and begin to treat all sorts of pulmonary diseases. I can get them in your bladder, colon, any area where we can address things topically. This creates a shot on goal, to use the soccer analogy or football analogy, to begin to develop a new therapeutic. And so this is a game-changing platform that we think is not going to just yield one drug, but literally hundreds of drugs over the course of the next few decades.
Okay, you use theoretical framework of biology in your research. Yes?
What?
You use theoretical framework from biology in your work.
We rely heavily on biology in our work.
Not biology. You don't use theoretical framework from biology.
We do work, and we use concepts of chemistry, biology, physics, and many different forms of engineering to create these types of medicine.
My question is if the theoretical thing that is for lengths bigger than nanotechnology scale, you can use the same form for nanotechnology scale.
I guess it's not clear what you're asking.
My doubt, I have a doubt about if you can use the same theoretical framework for nano scale and the another that is bigger than nano scale.
I still don't understand, I'm sorry.
Yes, I will write here in the chat.
I don't see the chat part.
Yeah, it's here.
Oh, can I see her? You're typing in Skype?
Yes.
It's not good. Yeah, you can use the same
Theoretical framework.
For what?
Então, I can explain. Framework.
But for what?
Different scale.
No, the scale here is important. I mean,
Scale nano or scale bigger than nano? No,
the scale is, so these little globular forms of DNA are typically about 50 nanometers in size. So and that's important because they're small enough that they can move throughout living systems, get to and encounter cells, and interact with the proteins on the surface of cells to be rapidly internalized. So scale is really important here. The properties of these structures are in part because they are nanoscale structures.
Okay, but in theoretical view, you have one particular theoretical view for nanoscale and another theoretical view for another scale that is bigger than nano? Or is the same theoretical framework?
I still don't get where you're going with the two different sizes.
Yes,
It doesn't make sense to me. Might be a language barrier problem. are two fundamentally different theoretical frameworks for nanoscale and bulk systems. In the sense that, look, one of the reasons nano is so interesting is that everything miniaturized in the nanometer length scale is different.
Okay, and is top bottom up?
Let me finish that statement because it's really important, I think, to the listeners. If you take any material, I don't care what it is, anything in nature, anything man-made, that is in bulk form, and if you shrink it down to the nanometer length scale, it will have different properties. Gold, which we work with all the time, is a beautiful example of that.
Yeah.
You shrink gold down to the nanometer length scale, it's no longer golden color. If it's a sphere that's 20 nanometers in diameter, it's red in color.
Yes.
If you change the shape of that particle, you can completely change all of its optical properties, and you can have one material that has any optical property you want within reason. So being able to structure materials in this length scale is really critical in terms of controlling their properties. Now, that's true of everything, including DNA, for that matter. If you rearrange DNA on the nanometer length scale, it has different properties, and I give you the example of the spherical nucleic acid. If we take the gold example, an easy way to understand this is that if we think of the properties of a material, there are what we call bulk atoms and surface atoms. With a large chunk of a material, the vast majority of the atoms that contribute to the properties of that material are in the interior, not on the exterior surface. If you keep shrinking that structure down to a smaller and smaller sphere, pretty soon you get to such a tiny particle that it's all surface. And those surface atoms have different properties than bulk atoms. They see things differently. Above them, we've got the environment. Below them, if they're a bulk material, we've got bulk atoms. The bulk atoms are surrounded above and below by the same types of atoms. So for that reason, everything, when shrunk down this length scale, will have different properties. And that's the exciting thing about nanoscience, that it's a whole new playground, a whole new field of study, because the field focuses on defining what those differences are. What does gold look like when you shrink it down to the nanoscale? Why is it that way? And then once you understand there's a difference, how do I use those differences to develop new technologies? That's the exciting part about this. Everything old becomes new simply through miniaturization.
Okay. And the process of your research for this subject that you are discussing here, you use the process top-down or bottom-up?
We use both. So top-down, of course, would be using lithographic tools, and we often make device components using conventional forms of lithography. But we also use a lot of bottom-up. Making spherical nucleic acids is a bottom-up process, where we are using effectively chemistry to produce lots of particles at scale. Bottom-up is great because it first of all gives you greater control, but you can also, if you use chemistry through bottom-up type of processes, you can do things at scale.
But it's more difficult to control the bottom-up process?
It depends on what you're talking about. I think it depends upon the system. For making spherical nucleic acids, it's a lot easier using a bottom-up process, chemical synthesis in this case, than it is top-down. But for making lithographically defined electrodes for sensor devices, top-down processes sometimes work a lot better than bottom-up processes.
Okay, and about this characteristics of the I can read here is to cross the blood-brain barrier. You think this is one good effect to introduce new medicine?
I missed the first part.
if the process that can cross the blood barrier in the brain,
Yes.
this is a good way to put new medicine, to use new medicine.
Oh, no, I think it's an exciting development. The fact that you can get these types of drugs to cross the blood-brain barrier is exciting for people that suffer from brain cancer. It may be very exciting for people that suffer from Alzheimer's disease, Parkinson's disease, and Huntington's disease, and many other types of ailments of the brain, maybe even treating strokes, for example. I think this is, again, just the beginning. And we're going to learn how to use this property to hopefully develop a lot of medicines that significantly improve human life.
But I read many times that this is the blood barrier is a very important natural defense of our body against many things. You think that he can use his equipment in very important way, but with security.
Yeah, but you're not going to use this with healthy people, right? So the difference between you and me, we want our blood-brain barriers intact, and there's no reason to do anything to try to get things across the blood-brain barrier. But for somebody who has a brain disease, this may be the only way of thinking about effectively treating them. And we don't have good drugs for Alzheimer's. We have very few good drugs for things like Parkinson's and Huntington's disease, and we have no drugs that will work well with glioblastoma multiforme, this form of brain cancer that I was referring to. So for many of those patients, first of all, oftentimes they're older. That's not universally true with the GBM example, but many of them are older. If you come down with one of these diseases, you can have a very grim prognosis. And so we need to have drugs, first of all, that can get to the brain if we want to treat them. And the fact that these cross the blood-brain barrier looks very, very attractive in that regard. For somebody who has glioblastoma multiforme, the average lifespan is about 15 months. So what we're trying to do is to create drugs that would significantly extend their lives. And so while there are concerns about issues like long-term toxicity and ultimately long-term side effects, that is not the main focus of somebody who has this disease. And frankly, if we could get something that would increase lifespan by several years, that would be considered a major advance in the field. If we could cure it, it would be fantastic, more fantastic.
Okay, and what is the bottleneck in the developing in your research? You can. It's always a combination of money and regulation. Right? It's not cheap to develop drugs. It's not cheap to do all the studies required to start commercially developing drugs. And so at the university, we try to pick what we think are the lowest hanging fruit, the avenues that will have the greatest impact for the number of resources that we have. And then we try to get companies, startup companies, to actually take those early studies and turn them into real drugs. And to do that, we have to raise the proper resources to do it. But we also have to be able to deal with all the regulations, the regulatory committees, government committees that regulate when we can run humans, how we run the human trials, and then ultimately how we interpret the results. Yeah, you think that nanotechnology was the pop star, and now the new pop star is graphene?
I missed the last part.
If you understand that the nanotechnology was a big pop star, and now the big pop star is graphene.
Well, I depends on field. I think the pop star changes from year to year, and depending on who you talk to. In the area of medicine, graphene is not relevant. It has very little impact in the area of medicine. I think that in this area, the pop star would be nanomedicine in general, but the silicon-nucleic acid example in particular.
Yes, I think nanomedicine is very important also to legitimate the research in general that is with nanotechnology applied to medicine field. I think it helps to legitimize nanotechnology in general.
No, I agree. I mean, look. Ivan talked about this. The silicon-nucleic acids with gold particle cores are actually used as labels in biodetection. They're part of a company called Luminex. And they're now used as labels in biodetection systems to track disease. They can very sensitively tell you whether or not a particular marker associated with a given disease is present. And one of the big applications is in looking for bloodstream infections, looking for markers associated with bacteria in your blood, because that's a sign of sepsis, which is a very deadly condition. And it's very important that you both accurately but also sensitively diagnose it, meaning that the faster you diagnose it, the earlier you can diagnose it, the better off the patient is, and the better chance of survival for the patient. This is a major technology that is not just a product of university-based research, but it's a product now used in half the world's top hospitals. And 20 years ago, 15 years ago, 10 years ago, you couldn't point to an example like that. But today, that technology is used everywhere. It's saving lives all over the world, and will continue to do so. Same type of thing with respect to clinical trials with nanomedicines. 10 years ago, there would be really no significant evidence of clinical trials. Now, many of the new drugs that are going into the clinic are nanomedicines. I've given you already several examples of silicon-nucleic acids, but other types of nanomedicines are also going into the clinic. And, I think that's exciting, because as you pointed out, those are tangible, significant outputs. It's also an important observation to make, because I think the general public and politicians expect things to happen very quickly, but in this field, you'll see diagnostics develop first because they have a shorter development cycle. The clinical or regulation in the clinic is not as severe as it is in the case of therapeutics, so it's a shorter time period through regulatory assessment. But then you'll see shortly after that the development of therapeutic tools, and that's what we're seeing in the field of nanotechnology. And I think you're going to see many new drugs that are either purely advances or purely from advances in nanotechnology, the silicon-nucleic acid examples being one of them, or the old drugs or existing drugs are made better through advances in nanotechnology, allowing you to deliver those drugs more accurately and selectively the different types of tissue types.
Do you believe that he will have a Nobel Prize in nanomedicine?
I think you'll have to ask the Swedish Academy that.
No, more easy ask you.
Look, it all depends. In the case of medicine, I think there are many examples of advances in science that have a very strong relationship with nanotechnology that are worthy of Nobel Prize. So I think you undoubtedly will see advances from nanotechnology recognized with Nobel Prizes. In the area of medicine, I think you will ultimately see, but it's going to take the development of a tool or a therapeutic that really changes the field in a very significant way, in a very profound way. I think we're on the verge of that in a variety of different areas, but only time will tell, and then only the Nobel Prize can lose it aside, right?
Yeah. You have a coleguinha with Nobel Prize?
That's right. So one of the things that our institute has done, which is great, is we've been able to attract the best and brightest from around the world to pursue their research efforts here. We moved Sam Soup here from the University of Illinois. We moved Milan Mursic from the University of Chicago. We moved Will Tell from Cornell. We moved John Rogers from the University of Illinois. We moved Fraser Stoddard, from UCLA.
Yeah.
He won the Nobel Prize last year. I had the party at my house. It was a very exciting event, and that was for making molecular machines. Very fundamental contribution, but a very important contribution, and one that changes the way you think about molecules and bonding and what can happen with molecules on the tiniest of scales. And so I'm certain that we're going to see a very large number of prizes and accolades going to advances in nanoscience and nanotechnology, because frankly in science that's where much of the action is. That's where much of the excitement is.
So the Nobel Prize is near you.
Excuse me?
The Nobel Prize is near you.
Well, a Nobel Prize winner is near me. Fraser Stoddard is one floor below me.
Yeah. So maybe the next.
Excuse me?
Maybe the next.
We'll have to see. It's hard to predict the future.
Yes, it's very difficult.
I could make a lot of money if that were the case.
Yeah. So I would like to thank you very much for your conference for us. I understand many things that I never understand before. So thank you so much. And when you have finished another project, maybe he can have another program to explain for us your new knowledge.
Okay, I look forward to it. I thank you for highlighting what we're trying to do here, and everybody for taking an interest in what I think is the most exciting field on the planet right now.
Okay, thank you.
Okay, bye-bye, thank you.